office hours. Today I'm really under the weather. I barely have enough energy to get through today's lecture, so I'm canceling office hours today, and hopefully on Wednesday I'll feel better. But Dr. Damianowski and I are going to have additional office hours this Thursday, so he'll post an announcement. Dr. Damianowski will have a Zoom office hours at 10 a.m. on Thursday, and then I will follow afterwards at 11 a.m. Okay, so we're going to finish up talking from the last lecture, finishing up the G-protein coupled receptor lecture, talked about vasodilation, and talked about how the relaxation of the smooth muscle cells surrounding arteries, how does that control? And so we talked about the initiating event will the parasympathetic nervous system cause the release of acetylcholine or bind to a G-protein coupled receptor. So I covered this in the last lecture, just kind of remind you. And we go through the usual steps. In this case, the effector is phospholipase C, which is going to cleave that membrane-associated molecule, PIP2, into diacylglycerol and N-acetyl triphosphate, or IP3. And that'll go and bind to the calcium-mediated channels of the endoplasmic reticulum, causing the release of calcium, which can then bind to and activate polymodulin, which in turn activates an enzyme called nitric oxide synthase, which will produce nitric oxide as a gas. It's able to cross membranes. It can cross through the membrane of the endothelial cells and then the smooth muscle cells, and then it'll bind to this nitric oxide, an enzyme that acts as a receptor, and it has enzymatic activity. So its enzymatic activity, which is guanyl cyclase, gets turned on when nitric oxide is bound to that enzyme. And that catalyzes the conversion of GTP to cyclic GMP. And that's an essential activator for protein kinase G, which will go and phosphorylate a variety of things that cause the muscle fibers to relax. And when that happens, you get increased blood flow into that artery. And we talked a little bit about angina and how we can kind of hack this system by taking nitroglycerin, which decomposes very rapidly to nitric oxide. So you kind of bypass all this stuff and then directly activate the guanyl cyclase, which will cause increased production of cyclic GMP and cause those muscles surrounding arteries. In this case, if it's angina, and muscles that the coronary arteries that are feeding the heart muscle. Okay, so then an interesting application. How many people here have heard of the drug Viagra? Okay, how many people have used it? Okay. So, interesting drug. It was initially developed as an antihypertensive medicine. And it does work somewhat. It does work a bit. It's not super great as an antihypertensive. But when they were testing it, they noticed, particularly in older men, that they had this curious side effect, and that they were getting erections. And so this sort of led to the development of this massive industry related to treating erectile dysfunction. And so at its peak, Viagra sales were around $2 billion. Now, over time, the patent has expired in various countries. Now it's completely expired. So, Pfizer is only making about $500 million a year now off this. So they're still making quite a bit, but other off-label brands are available, and it's still now in the neighborhood of about $3 billion, and projected over the next 30 years, because people are getting older, to exceed $6 billion in sales. So a very lucrative drug. How does it work? Well, it basically binds to and inhibits this enzyme called phosphodiesterase, PDE. So in the last slide, we talked about cyclic GMP, and that was being an important cofactor, or an activator, second messenger to activate protein kinase G. Well, what happens is phosphodiesterase can break down cyclic GMP to GMP, and that can no longer activate protein kinase G. And so that would actually cause constriction of the blood vessels. How does this relate to erectile dysfunction? So a little penis anatomy, I guess, that when there is an increased blood flow into the penis, what happens is blood flows into these tissues called the corpus cavernosum. And so that causes the penis to engorge, and that in turn will cause constriction on the vein. And so what happens is blood is going in, so the penile vein gets compressed and then blood can't come out. So if there's a way to basically increase blood flow into the penis and then maintain that blood flow, that's the whole thing. So how does this work? All right, so you have a dirty thought, and this causes activation of the parasympathetic nervous system, causes a release of acetylcholine, and that is going to cause all those effects in the vascular endothelial cells, in this case the penile artery. And so that in turn leads to all those events that we talked about in terms of phospholipase C and calcium and nitric oxide, and ultimately you're going to get activation of the guanyl cyclase enzyme because nitric oxide activates it. So you're converting GTP to cyclic GMP. That's going to increase the activity of protein kinase G, causing phosphorylation of a bunch of stuff to cause the muscle relaxation, more blood flows into the penis, and you know the rest. So the problem is that this process is competing against a process that's countered by phosphodiesterase 5, PDE5. And so the cyclic GMP is broken to GMP, and then basically this is stopping activation of protein kinase G. So what Viagra does is it basically inhibits this process. So it artificially maintains the levels of cyclic GMP. It doesn't get broken down. And therefore it artificially stimulates persists in stimulating protein kinase G, and that causes relaxation of the smooth muscle cells, and hence increased blood flow in the penis. All right, so that's how it works. So this is, obviously a lot of people are using it, and I guess this is my public service announcement. You might think, hey, take one little blue pill, maybe it does this, 2 must be better. Well, I would be a little cautious about doing that. So there are side effects to this. Now, if you're already on antihypertensive medicine, this is going to exacerbate that. It can cause actually a very strong, it can cause a quite pronounced drop in your blood pressure, which could lead to, ironically enough, a thing like a heart attack. If you drop your blood pressure too low, the heart can't pump enough, or it doesn't get enough blood back, so you're basically systemically dropping all your blood pressure. The heart itself is maybe not necessarily getting as much blood as it needs in a condition. It can lead to a heart attack. Likewise, if you're not directing enough blood flow up to your brain, because you're basically causing blood to dissipate all throughout your body, this could lead to a stroke. It can also has indirect effects in terms of cochlear cells and related to hearing, and it also can affect the rod and cone cells in your retina such that it causes them to not respond properly to light, and so it can lead to what's called cyanopsia. Cyanopsia, that's it. So basically everything looks like blue because you're not activating the rod cells that let you sense light properly, so it's biased towards blue. And so here, there's another thing. So priapism. And when I first was looking at side effects and I saw priapism, I had no idea what it was. And I looked it up and I went, oh. Okay, so it's basically an erection that doesn't go away. And an erection that lasts for maybe an hour, 2 hours, you're starting to get into some serious problems. Why? Because the blood that's flowing in has to be returned and reoxygenated. So what happens is there's only so much blood that can flow into the penis, and it now becomes depleted of oxygen over time. And without the restoration of oxygenated blood, this can lead to the death of the tissue. It's called gangrene. So by preventing an influx of fresh blood, it can lead to the destruction of penile tissue. The only way to treat this is actually go to the hospital, they put a big needle in, and then they actually physically remove the blood. So just think about that. The next time anybody recreationally wants to use Viagra, that might make you think twice. Okay, so on that pleasant note, we're going to move on to get a little conversation here. Okay. Okay, so we're going to talk about the next category of really big category of receptors, cell surface receptors, called receptor tyrosine kinases. We'll talk a little bit about their structure and how they get turned on, and some of the mechanics of how there's a number of events that happen with the receptors themselves, and then there's a whole bunch of downstream proteins that can transmit these signals. We'll talk about these adapter proteins. We'll bring up another small monomeric G protein called RAS. It plays a really important role in a lot of signaling that's messed up in a lot of cancers. And then we'll talk about this category of human epidermal growth factor receptors called HERs and their involvement in human breast cancer. And then finally, we'll also talk about these kinases that get turned on and are implicated in growth and mitogenesis. Okay, so what are the types of things that turn on these receptor tyrosine kinases? Many things. Most growth factors, not all, but many, work through them. And insulin also works through receptor tyrosine kinase, or RTK. I'm talking in a little bit more detail about the structure. It's a lot different. For example, a G protein coupled receptor has many transmembrane domains. This has a single one. And as with most cell surface receptors, it has an extrinsic component which binds the ligand, and obviously the transmembrane domain, and then it has a cytoplasmic portion which has intrinsic tyrosine kinase activity. So that's it has enzymatic activity that puts phosphate groups on tyrosine messages. And then we'll also talk a little more detail about how receptors don't work. Rarely do they work on their own. They have to associate with another receptor to form what's called a dimer. And then we're going to talk about, once that happens, what are some of the other proteins that help transmit the signal from this activated receptor to downstream proteins. So we'll talk about adapter proteins. Then there's the involvement of a G protein called RAS. And then we'll talk about how, when there's many aspects of this whole signaling process that, when they get messed up, are the underlying cause of many cancers. So last couple of lectures, we talked about G protein coupled receptors. And so as I said here, it's got that undulating snake type of structure, activates trimeric G protein complex, and that leads to downstream events. It could be activation of adenyl cyclase, producing cyclic AMP, and so, it can cause activation of certain kinases. The kinases in turn can activate things like transcription factors. We talked about CREB, for example, and that in turn can go and activate or potentially repress expression of certain genes. So we're going to go through a lot of the details here, but just to give you a kind of overall broad brush stroke of how it works. With RTKs, they work a little differently. So usually the binding of ligand to the receptor will cause dimerization, which we'll talk about in a little bit more detail in the next couple of slides. And you're going to usually lead to recruitment of various adapter proteins, which aren't shown here. A small monomeric G protein called RAS can be activated. And one of the key things here with RTKs is that it's a sequential phosphorylation cascade. So one kinase phosphorylates another kinase, which phosphorylates another kinase. Which at any point, these kinases can go off into various myriad of directions, or they may culminate with activation of a transcription factor via phosphorylation. All right, so that's kind of just, remember, we talked a lot about this. There's some differences. So the structure of this is quite different from the structure of that. The activation of this receptor will be associated with the activation of those trimeric G protein complexes, so G alpha S and all that kind of stuff. This, the activation is dependent on kinase activity in the cytoplasmic domain, which we'll talk in more detail in the next couple of slides. And that's going to cause recruitment through some intermediate proteins, which we'll talk about, to activation of a monomeric G protein. So So there's some differences here to take note of. So as I mentioned, that the binding of a ligand to the growth factor receptor will cause its activation. So the activation occurs through a couple of different steps. First off is one ligand binds to one receptor. In some cases, not all, and we'll talk about some exceptions to this rule. Another ligand may bind to the other receptor, and that triggers their association together. So they dimerize, two of them associate. And that dimerization is important for activating the intrinsic kinase activity. So this is the outside of the cell, there's our single transmembrane domain, and here's the cytoplasmic portion with the kinase domain associated. When these two proteins, two receptors, bind together and dimerize, it causes activation of the kinase. And then what they do is they autophosphorylate, trans-autophosphorylate. So this kinase phosphorylates that one, this one phosphorylates that one. So there's certain tyrosine residues within the cytoplasmic domain that get phosphorylated. So the kinase, this one phosphorylates that one phosphorylates that. So they kind of go back and forth, and they slather all these phosphate groups on tyrosine residues, and that actually acts as a signal to cause a recruitment of what we're going to call adapter proteins. So the phosphotyrosines act as a signal or a docking site for these downstream adapter proteins, which contain SH2 domains, which I'll explain in a little more detail. So we've already talked about how we get activation of the receptor. Now it's all phosphorylated on tyrosine residues, and they're going to be recognized, those phosphorylated tyrosine residues are going to be recognized by an adapter protein via a domain called SH2, which stands for Sarc homology 2 domain. The Sarc is an acronym for sarcoma, it's a type of cancer. So, as it turns out, many adapter proteins, and there are at least 100 in humans, contain these Sarc homology domains. So they're like it's sort of like an interaction domain. That's the best way to classify it. They act as a bridge connecting one protein to another. So a little bit of background, where does the name Sarc come from? It actually goes back over 100 years. In this case, a researcher by the name of Francis Peyton Rouse had been doing experiments and trying to understand what causes cancer. And so he had taken extracts from a chicken tumor, a sarcoma, and he had killed all the cells in there, and then took that extract and he injected it into healthy tissues, and they all developed tumors. And so this was the first example of some kind of infectious agent that could promote cancer. This is greeted with a lot of skepticism in the early 1900s. And so if you look here, when he did this experiment in 1911, he was finally awarded the Nobel Prize. Unfortunately, he's still alive in 1966, in part because nobody believed him. They felt that he hadn't completely killed all the cancer cells, and there were still a few residual cancer cells, and you're just transferring cancer from one animal to another. This is not the case. It was actually a virus, a retrovirus. It was now called the Rouse sarcoma virus. And so as we move, so they knew that the virus was causing the cancer. The question is, how does the virus cause the cancer? And we had to get into the 80s when we started to have a little bit, still rudimentary, but some molecular tools to identify what kind of genes are found in viruses. In this case, when these individuals had, Bishop and Vermis, were able to identify the genes encoded by this retrovirus. And when they looked at the gene, they found that it encoded for a protein that had kinase activity. That's the first thing. And then it had these certain protein sequences and these Sarc homology domains, SH2 and SH3. And then they found that these domains were found in many other proteins. And so the bottom here is actually what they found in the virus. And what had happened is the virus had integrated into the chicken genome, and then it had popped popped out. And when it popped out, it took a piece of the kinase from the chicken genome, but it missed a piece. So what here, V start stands for viral SARC, and C start stands for cellular SARC. So this is the normal version of the gene that's found in the chicken genome, and there's versions of this found in us as well. And what had happened when the virus had popped out of the chicken genome, it failed to fully take the whole gene from this particular kinase. And so it was missing the carboxy terminus. And this is important because it's phosphorylated by a certain enzyme, a kinase, which I'm not going to make you remember yet another acronym. And by phosphorylating the carboxy domain, this actually inhibits the kinase activity. That's in the normal version of the gene found in the chicken genome, and it would be found in our genome. A human version is found in us. In this particular viral SARC, that is missing. And so, therefore, the kinase is turned on all the time. And so, in this case, by having that kinase turned on without an off signal, it can phosphorylate a lot of steps, a lot of other kinases that are involved in mitogenic signaling. And that's what's promoting the cancer, just by having a constitutively turned on all the time kinase phosphorylates a number of downstream proteins, which we'll talk about in a minute, that leads to the hyperactivation or hyperactivation of mitosis and cell growth. So, so the normal version is C-SARC, cellular SARC. The viral version is V-SARC. Pretty well the same, except the viral version is lacking this regulatory domain at the end. It can't be phosphorylated, and therefore the kinase activity here can't be turned off. The other important point to note is that this particular protein SARC also contains these SH SARC homology domains. And so they're...
Sorry, so just to confirm, so if you were to be infected with the virus, you still have your own cellular sarcoma that's working normally, that's fine. You just also have this other one.
So if you were infected with this virus, good point, you still have your own cellular version, which could be regulated, but on top of that, you're going to have this viral version, which will be made active all the time. Okay, so before I talk a little bit more about the SARC homology domains and what they do, I'll introduce one other signaling protein, which is RAS. So this is a small G monomeric G protein. GTase switch protein, as it's referred to. Basically, we've talked about these types of proteins in the past, that in their inactive state, they're bound to GDP, and in their active state, they have GTP bound. And that these particular monomeric G proteins generally need a lot more help in terms of, we talked about the G alpha S and the trimeric G proteins. Monomeric G proteins tend to be more reliant on things like GAPS, guanine nucleotide exchange factor, to help remove the GDP and allowing GTP to bind to make it active. And then they also rely on these accessory proteins like GTPase accelerator proteins or GAP proteins, which help hydrolyze the GTP to GDP. So remember, like in any signal transduction pathway, you have to have that on switch and you have to have the off switch. The on is with GTP bound, the off is with hydrolysis of GTPs, flicking the switch down to GDP. As it turns out, RAS is very dependent upon these things working properly. And in 20 to 30% of human cancers, there's a mutation in glycine 12, which actually prevents RAS from binding to a gap. So what happens is it can bind GTP, but it can no longer bind the gap protein, and that can't hydrolyze GTP to GDP. And as a result, the RAS protein is turned on constitutively. So this is what they refer as the RAS-D. I think probably D stands for dominant, meaning that if you have this mutation, it will lead to a permanently active RAS protein. It's going to turn on all these signaling pathways that are related to cell growth. Okay, so now we're going to kind of put all this together. We already talked about how you have a ligand and it binds to the receptor, causes the dimerization. And then the autophosphorylation of the cytoplasmic domains are tyrosine residues. And now this is going to act as a signal for an adapter protein called GRAB. Kind of makes sense because it kind of grabs onto things. But it technically means growth factor receptor bound protein, which also makes sense. So it's an interesting protein in that it has no activity itself. There's no kinase activity or any other enzymatic activity. It simply helps link the activated receptor to other proteins that do things. And so it has a domain called SH2 or sarcomology 2 domain, and then it also has another domain called sarcomology 3. And so these are important for facilitating interactions between the activated receptor and downstream signaling proteins. So when we have activation of the cytoplasmic domain, so now we have tyrosines that are all phosphorylated, they're going to be recognized by GRAB2 via the SH2 domain. So it binds to phosphotyrosine residues that are on the activated RTK, that are facing into the cytosol. And that, in turn, causes a shift in the SH3 domain, such that it's going to bind proline residues in another signaling protein called SOS. So, GRAB, it's got two pieces. SH2, that's going to recognize phosphotyrosine. And then it's got SH3, which is going to recognize proline residues on SOS. So these are two interaction domains. That's what SH2 sarcomology domains do in a variety of signaling proteins, is they help facilitate protein-protein interactions in all these types of signaling events, usually associated with RTKs. So at this stage, what happens is you've got activated RTK. GRAB is latched onto it via its SH2 domain. The SH3 domain reaches over and grabs onto SOS, and now SOS is going to bind onto this little thing here, this bluey thing, is RAS. So that's that monomeric G protein. It's got a lipid anchor, which connects it to the plasma membrane. So at this point, what's going on? We've got now everything's all linked together. The RTK, the GRAB, the SOS, the RAS. You got the whole kitchen sink in here. What happens now? So SOS, once it's being bound to this complex, will turn on an activity, which is, again, guanine nucleotide exchange factor. So it activates its exchange activity, which will allow it to remove GDP from RAS and allow GTP to bind. And at this point, now RAS is fully active and it has a change in its structure that it dissociates from the SOS. So now it's activated and it can go on to promote the phosphorylation and activation of other kinases. We'll talk about RAF. Ultimately, it's going to lead to the activation of a family of kinases called the mitogen-activated protein kinase, which is exactly what it describes what they do. They are kinases that get activated in response to mitogens. Okay, so a little bit more detail. It's a little hairy here, and I try to keep it down to a little more as best I can. This is, again, sort of the superficial covering of how this works. I'm trying to keep, the textbook loves introducing more detail, which I don't think is really that helpful, but let's try to simplify. So we have activation of that RTK that's happened before. SOS has helped remove GDP, allowing GTP to bind. Now that's going to move along, and it's going to interact with a protein called RAF. And so RAF is a kinase that is held in an inactive state via an inhibitory protein called 1433. I didn't come up with these names. And so it binds to certain phosphorylated groups on this kinase. What happens is RAS will help recruit RAF up to the plasma membrane. And it, through some other mechanism, which I'm not going to make you memorize, promotes a dephosphorylation of RAF. And this inhibitory protein 1433 is kind of unlatched. And that in turn allows the activation of RAF. And then at this point, what happens is you get GTP hydrolysis on the RAS, and this dissociates RAS from RAF. So now activated RAF can dissociate from RAS, which is now inactivated, and that in turn will go on and phosphorylate the next kinase in the chain called MEK. MEK in turn will then phosphorylate another kinase called MAPK, or mitogen activated protein kinase. And then the MAPK can go on and phosphorylate many things. In quite a few cases, it can go into the nucleus and phosphorylate and activate transcription factors. And those transcription factors can regulate things like genes involved in cell cycle, maybe like cyclins, for example. Cell growth or cell differentiation. So, the idea is understanding it's a sequential phosphorylation of a kinase, which phosphorylates a kinase, which phosphorylates a kinase. And so, when this stuff was all being discovered, people came up with different names for it just to make life really difficult. And so, they started with the bottom one, calling it a MAP kinase. And then they said, well, what's the kinase that phosphorylates that? And that's a MAP kinase kinase. And what's the kinase that phosphorylates that? A MAP kinase kinase kinase. And then they thought, well, that's kind of really awkward. So they kind of came up with different, just tried to agree on their names like RAF, MAC, and MAPK. So what is important to remember is that what are the order of events from the RTK to RAS to RAF to MAC and MAPK? Most of these kinases are what we call serine-threonine kinases, with the exception of MEK is a dual kinase. It can phosphorylate serine and threonine in addition to tyrosine. So, I try to, not have too much superfluous information. This is what I think is the bare bones for what you need to understand. And I think the next step is to, okay, how does this all relate to something that maybe we can relate to, like cancer? And so, what has been discovered over the last 40 years is that many of those signal transduction pathways that I've talked about, be it RTKs or RAS or those MAPKs or those MAPKKs or KKKs, is that there's mutations in these genes that encode these proteins that lead to those particular signal transduction proteins in a constantly on state. So this is, these gain-of-function mutations are oncogenic. So in some cases, though, we have mutations in genes that encode for proteins that act as the brake to stop this. So these are what we would call tumor suppressor genes. So they are normally responsible for turning off the signal. And so it's usually loss-of-function mutations. So they're losing their normal activity to suppress this pathway, and that can allow the pathway to go unchecked and be turned on at a higher level than you would want. So we have two types of mutations, gain-of-function mutations. So that means that the mutation leads to that particular signaling protein, be it the RTK or one of those kinases, being turned on all the time. And it can't be turned off. So those are oncogenic mutations, usually gain of function. Or there's a mutation in one of the brake type of proteins. So those would be responsible for suppressing the potential for new tumors to form. So by removing the brake, now that can also lead to uncontrolled cell growth. So what's shown here is basically all the members that we talked about, from RTKs to adapter proteins to RAS to RAF, MEK, MAPK. All the ones that are shown in green are potentially proto-oncogenes. So that means if there's mutations in them that could lead them to being turned on too much, it could potentially, not always, but in many cases, can contribute to cancer. And what's shown here in red is a mutation in a tumor suppressor gene. So this one is GTPase activating a gap protein called NF1. So we talked about RAS, for example. We can have two situations where RAS can be turned on. So it could be mutation like glycine in position 12, the RAS-D version. So that means that it cannot recognize or bind to the gap, in this case NF1. So that leads to it always having GTP bound to it. The opposite, or the alternative way of leading to RAS being turned on too much is that there's an inactivating mutation in NF1. So therefore, it either cannot interact with RAS or no longer has the GTPase activity. So in that case, in either situation, having GTP bound to RAS and not being able to hydrolyze it is going to lead to chronic RAS activation. As I said, 20 to 30% of many human cancers are associated with chronic RAS activation. Okay, so maybe we'll now just focus a little bit on the receptors. So many of these RTKs, as I mentioned, are dependent upon the dimerization of receptors. So one class of receptors are the human epidermal growth factor receptor, EGF. So it's also called HER2. And we'll talk about that in a little bit more detail when we talk about breast cancer. So you can have certain types of mutations in which there's a mutation, for example, that within the transmembrane domain, so in this case availing to glutamine, causes the receptor to dimerize in the absence of any ligand. It just spontaneously dimerizes and causes the activation of the RTK domains. So now, in the absence of any ligand, it is leading to chronic RTK activation. So this is called the new oncoprotein. It was identified in a brain tumor, so neuroblastoma. Or you can also have a situation where there's a mutation that causes the deletion of the extracellular ligand binding domain. And so that leads to the EGF spontaneously dimerizing and activating even in the absence of any ligand receptor interaction on the outside. So just, again, both things cause spontaneous dimerization, they just do so in slightly different ways. So this one, you still have the extracellular domain present, but the mutation within the transmembrane domain causes them to artificially dimerize. In this case, removing most of the extracellular domain causes a conformational change. So they don't have that big, bulky extracellular domain, and so they're much smaller, and they can dimerize more easily. And again, this is leading to chronic activation of the cytoplasmic kinase domain. And this is an herb with, if I vaguely remember, something to do with blood cancer, erythropoietin. Don't quote me on it. I can't remember, so we'll test on that. So, okay, so these human epidermal growth factors, there's 4 of them. HER1, HER2, HER3, and HER4. And so these can form, in some cases, they can form homodimers. So you get HER1 when bound to EGF on one receptor can cause, and then EGF binding on the other receptor can cause the dimerization, just what we talked about earlier. And so you can also get combinations of HER1 with HER2, HER3 with HER2. So these are kind of mixed dimers, heterodimers as they call them. The interesting thing about HER2 is that it actually does not bind to EGF, but it can be activated when bound to another HER family member like HER1, HER3, or HER4 when they abound EGF. So you only need one EGF present. It'll bind to either HER1, HER3, or HER4, and that will cause the heterodimerization with that other HER isoform with the HER2. And that leads to the activation of the RTK domain. So, this is of interest because in many, 25% of breast cancer patients have some kind of a genetic event that leads to increased HER2 expression. It could be duplication of the HER2 gene. It could be some alteration that leads to excessive production of the HER2 receptor. And so normally what happens is we have a small amount in a normal cell of the HER2 receptor at the plasma membrane. But in a cancer cell, now you have much more HER2 receptors that are up at the plasma membrane. And this is, and as a result, activation of this receptor can occur at very low concentrations of EGF. There's just so much HER2 up there. And so, noting that there's such a high level of HER2 on breast cancer cells, this led to the development, the first actually development, of a monoclonal antibase therapy. And this led to the drug called Receptor. That's the antibody that was directed towards the extracellular domain of the HER2 receptor. And it actually can be quite effective in treating HER2 positive breast cancer. So how does it work? Well, so remember just what I talked about, that you can have EGF binding to either HER1, HER3 or HER4, which can cause the dimerization with HER2. And that's going to lead to the pathways that we talked about. Don't worry about this pathway, let's just focus on what we talked about. SOS, RAS, RAF, MAPK, will go in phosphorylated transcription factors are associated with cell proliferation, cell survival, or perhaps even things that can make a cell promote change into a state where it's more invasive, it wants to move more, or it's going to secrete factors that can cause the production of new blood vessels to come towards that particular cell. And it can then allow an escape route through blood vessels. This is called metastases. So the Herceptin antibody, what it does is it can do a couple of things. The one that is kind of easy to understand based on everything we talked about is that it can bind to the extracellular domain of HER2. And it basically physically blocks its ability to associate with other HER isoforms, either HER1, HER3, or HER4. Or it can even also prevent HER interactions. Sometimes you get spontaneous dimerization of the HER2 receptor. Now, according to the company that makes it, they claim that it does all these other things. It could be true. I think it's predominantly working through this mechanism, but there is some evidence, perhaps, that the HER2 receptor can be potentially susceptible to a protease, an extracellular protease, which cleaves off the extracellular domain. And when that happens, remember we talked about if you remove the extracellular domain, in the other case we're looking at a certain form of cancer with a genetic mutation, but this protease enzyme, by cleaving off the extracellular domain, can cause spontaneous dimerization of the HER2 receptor, leading to its inappropriate activation. So there's some evidence that the Herceptin antibody, that's what this little thing is, there's the antibody, can bind to and prevent the protease from cleaving off. So far I've talked about two potential mechanisms that Herceptin can work. One is by binding to the extracellular domain of HER2, preventing it from dimerizing with other HER2 isoforms. That's a key part for activating it. Secondly, that it can bind to the part of the extracellular domain and prevent the protease from coming in and trying to cleave off the extracellular domain, which can lead to the autoactivation of the cleaved HER2. Now, there's also, as an antibody, it can be potentially recognized by immune cells. So when you have HER2 bound with the antibody, it can cause the recruitment of immune cells, which will recognize this antibody and can potentially mount a cytotoxic response, which would initiate apoptosis of these cells. We'll talk about apoptosis in the next lecture. And there's also some evidence that the antibody binding to the extracellular domain can cause basically endocytosis of the trigger endocytosis of the HER2 receptor. So it gets endocytosis perhaps degraded in the lysosome. So you're removing, causing artificial destruction, internalization and destruction of the HER2 receptor. So again, 4 potential mechanisms that it could work. One is preventing the dimerization. One is preventing the proteolytic cleavage of the extracellular domain. Third is by causing an immune response to mobilize immune cells. It could be cytotoxic T cells to recognize this as foreign and cause a apoptotic response, which we'll talk about in the next lecture, or that the antibody triggers basically something akin to receptor-mediated endocytosis, something like that. The other thing that they have done is they've modified Herceptin. It's called Kadcyla, and so they link Herceptin to a cytotoxic drug. And so, in part, when this happens, we've got the drug. When it gets internalized, it can actually trigger the death of the cell. So that's even actually more potent as a, because if you were to give a drug, so a lot of the problems with chemotherapeutic drugs is they're not particularly specific to cancer cells. Now, they may work by targeting cancer cells that are highly proliferative, that the DNA is dividing very readily. That's where DNA is susceptible to certain drugs that will modify and mess up dividing DNA. So highly proliferative cells like cancer cells are susceptible to that. But you have healthy proliferative cells that also get adversely affected. So that's why when people are in chemotherapy, they can have a lot of GI discomfort because they're killing off a lot of your intestinal epithelial cells. Your hair cells can be affected, so you start to lose hair. So in this case, it's a magic bullet. You're actually targeting the chemotherapeutic drug to the cancer cells that are highly expressing this HER2 receptor. So therefore, it's a much more effective treatment. And I will finish there.